broadly neutralizing antibodies plus immune modulator may delay HIV rebound. More than half of people who received a pair of broadly neutralizing antibodies, BNAbs, plus the immune-modulating drug N803, Anktiva, had delayed viral rebound or maintained a low or suppressed viral load during antiretroviral treatment interruption, according to study results presented at the 13th International AIDS Society Conference on HIV Science, IAS 2025, among 24 participants who stopped antiretroviral therapy, ART, 58% did not meet the criteria for restarting treatment at 6 months. Nearly a third did not need to resume treatment after a year, and one is still off antiretrovirals after more than 30 months. With follow-up ongoing, Professor Marina Caskey of Rockefeller University reported, "So far, what we are most excited about is that we see among participants with delayed rebound that they show different patterns of rebound dynamics, and some of these participants maintain low-level viremia for a prolonged period of time," she said. ART can keep HIV suppressed indefinitely, but the virus inserts its genetic blueprints into CD4 T cells and establishes a long-lasting latent viral reservoir that is nearly impossible to eradicate. Cure researchers have tried many different strategies, including kick and kill, which aims to reactivate dormant HIV and flush the virus out of hiding, known as latency reversal, while boosting immune responses to fight it. People living with HIV normally produce antibodies against the virus, but these mostly target parts that are hidden or highly variable. However, a small proportion of individuals naturally make BNAbs that target conserved parts of the virus. Manufactured versions of these specialized antibodies are being explored in HIV prevention, treatment, and functional cure research. Drugs that stimulate immune function could potentially help BNAbs work better to control HIV. N803, being developed by biotech company ImmunityBio, is an interleukin-15, IL-15, receptor superagonist that activates natural killer, NK, cells and CD8 killer T cells and enhances CD4 helper T cell activity and proliferation of memory T cells. The U.S. Food and Drug Administration approved N803 as a treatment for bladder cancer last year. It is being studied for several other types of cancer in combination with immune checkpoint inhibitors, which unleash T cell activity. The drug is also being explored in HIV cure research. In addition to stimulating immune cells, it appears to have latency-reversing properties, suggesting it might both kick and kill. In 2022, Professor Timothy Shacker of the University of Minnesota and colleagues reported that people on ART who received N803 experienced proliferation or activation of CD4 and CD8 T cells and NK cells. HIV transcription in memory CD4 cells initially increased, followed by a persistent decrease in the viral reservoir. Last year, the researchers reported that people who received an infusion of NK cells from a relative plus N803 saw a moderate decrease in HIV-producing cells in their lymph nodes. This study had some limitations which have implications for patient safety. The participants were not tested in advance for sensitivity to the BNAbs, meaning they might have had pre-existing resistance. However, the PhenoSense test used to assess resistance is not a very good predictor of viral rebound, Caskey and other speakers in the session noted. Some apparently resistant viral variants can still be highly responsive, and relying on the assay could exclude some good responders. An additional concern is that the criteria for restarting antiretrovirals, for viral loads above 1,000 with no reported upper limit, could raise the risk for disease progression and HIV transmission. What's more, there was no control group receiving the BNAbs alone, making it hard to say how much benefit N803 added. In the Rio study, reported at this year's conference on retroviruses and opportunistic infections, 3/4 of men with early treated HIV who received the same 2 BNAbs did not experience viral rebound 5 months after discontinuing antiretrovirals, and a third were still in remission at 18 months. The FRESH study, which enrolled women in Africa who started antiretrovirals during acute infection, found that 30% of those who received 2 other BNAbs, BRC07-523-LS and CAP256V2-LS, plus the immune-modulating drug visatalimod maintained viral suppression without antiretrovirals for 12 months. Visatalimod, a toll-like receptor 7 agonist, activates NK cells and T cells, and may also have latency reversing properties. Here too, there was no control arm receiving the bNAbs alone to tease out the added effect of visatolimod. The ongoing ACTG A5386 study is a randomized trial that assigns participants to receive 2 bNAbs, BRC07-523LS and 10^1074, plus N803 or the superagonist alone, but again, there is no comparison group assigned to just the bNAbs. We are now trying to understand what's involved. What are the underlying mechanisms? Caskey concluded. How much of it may be related to residual antiviral activity? How much of it may be modulation of host immune responses with perhaps associated fitness costs to the rebound viruses? And the big question for this study is, will we be able to determine if N803 played a role in the effects we are seeing? It will be really nice to compare across all of the studies, and I hope that we will be able to do so.